So now we are going to see the discussion on like the secondary structures. Okay, secondary structures of the protein. So here you can see so you are seeing like primary, secondary, and tertiary quaternary structures, right?
Yes, sir.
Sir.
Yes, sir.
All right. So you know what is mean by primary structure, right?
Yes, sir.
So in primary structure, you will have like only amino acid sequence, and the secondary structure it describes about like alpha helix or like beta strands, right? So number of like you have like secondary structures. You have like alpha helix, beta strand, and the loop region. All right. So all are coming secondary structures. And then tertiary structure. Okay. So combination of these secondary structure. Okay. So combination of like these secondary structure will make the tertiary uh, whole. Okay. So then like several combination of these tertiary structure, which is making a quaternary structure of a protein. Okay. So some protein may be good enough to function just with a single polypeptide chain. Okay. So some protein will be made of only a single lean polypeptide chain. Okay. So some protein may be this single polypeptide chain folded into two different domains also. Okay. So single polypeptide chain, entire polypeptide chain may make like a global single domain, or it can make a double domain, or even it can make multiple domain. It will be just single polypeptide. Okay. And some proteins, it may require two polypeptide chain. Okay. So it will be a dimer or trimer or tetramer. Okay. So different octamers will be there. Okay. So depending upon the protein, you will have multiple polypeptide chains present in that protein. Okay. So just now we saw the example of myoglobin and hemoglobin. Okay. So in case of myoglobin, you require only one polypeptide chain, whereas in hemoglobin, it requires four polypeptide chain. Okay. So whatever that I have displayed here is represented by the hemoglobin only. So you have like a four chains in this in order to perform the function. Whereas in myoglobin, it requires only single polypeptide chain. Okay. So in case of this hemoglobin, you can call it as a homopolymer. Okay, it's a homooctamer or homopolymer because all the four chains are same amino acid only. All the four chains, four polypeptide chains, will be same amino acid. Alright. In case of ribosome, you will have like a number of polypeptide chains, and each polypeptide chain will be having different sequence. You got it? You are getting or not?
Yes, sir.
Yes,
Yes,
sir.
sir.
Or Vishal, do you understand?
Yes, sir.
So can you say what is in ribosome? Ribosome? Come on, Vishal.
Sir, it's a quaternary polypeptide, quaternary structure of polypeptide.
I'm asking about ribosome. Yes. Arrangement of ribosome. What, Mr. Vishal? What's the difference between hemoglobin and ribosome? Anyone else? Marali?
Yes, sir. Hemoglobin, it has four polypeptide chains. All of them have the same amino acid composition. Whereas in ribosome, there are multiple polypeptide chains, each having different sequences.
Very good. So I told you hemoglobin as a homopolymer. Homopolymer means all the polypeptide chain will be having same sequence. Whereas ribosome is not a homopolymer. Each polypeptide chain sequence is different. Okay, that's the thing that you should understand. Okay, so you see the diversity of the protein. That's what I wanted you to understand. See the diversity. Okay, so some protein just a single polypeptide chain, it is carrying biological function. Okay, single polypeptide chain, single domain. Single polypeptide chain, multiple domain. Still it is carrying function. Okay, some protein it requires double polypeptide chain, dimer. Okay, dimer, trimer, tetramer, whatever. Again, it is carrying function. Okay, and some polypeptide chain, it may be a homopolymer or heteropolymer. So it depends upon the protein. So you have like a variety of protein, variety of combination. So diverse structure we have, diverse function we have, okay, diverse arrangement, globular arrangement. All right. So if you go to the PD site, all right. So just download the structure and superimpose each structure and see how far it is like matching, right? How far it is overlapping. So definitely there will be difference, right? So that indicates like the diverse arrangement of like each protein. Okay. So how you are getting this different variety of shape, variety of structure, and also different kinds of function? How the polypeptide know that I have to make only this kind of fold, or I have to make only this function? So who is dictating, right? How the polypeptide itself it is folding in a native fold, right? So what is mean by native fold? Subhas.
Sir, it's like a true form of a protein.
Right, the original form, right? How the polypeptide chain know the native fold? Who is instructing, right? So whenever the polypeptide chain is like synthesized, right? So the tRNA, each tRNA is keep on bringing the amino acid, right? It is coining one by one, and it is making a long polypeptide chain. And at the end, once the polypeptide chain is like completed, so it will terminate the reaction, and the polypeptide chain it will go to the destiny. Once it is reached the place, right, it's going into the native fold, and it is starting to work. So who is giving this instruction? So each and every second, the proteins are being synthesized, and the proteins are being folded. and the proteins are continuously doing the function, right? Who is giving instruction? So the important thing is that there is something important information is buried. Okay, it is like lying in the sequence, in the primary structure, right? Each and every protein, it is coming from the sequence of the protein only, right? So this primary structure does carry the important information about the three-dimensional structure. It is carrying important information about the function of the protein. Okay, so the primary structure is like very important. Okay, so that's why people are like wondering whether we can make um, three-dimensional structure as well as a functional prediction directly from the sequence, right? So nowadays we require like experiment. You need lab facility, right? First you have to determine the structure of the like protein, and then you have to go for the functional analysis. So which is like a long way in a lab to do, but so all this information, the structure as well as the functional information, already buried in the sequence. Why not we gather that information directly from the sequence? That's what people are trying. But still, we are not able to understand this mechanism. We are not able to extract this information, this useful information directly from the sequence. So people are still keep on working. Okay, so that is resulting like several programs. Okay, so now we have a variety of programs which can build the model from directly from the sequence. So whether you have a model or you don't have the model, whatever. So from the sequence, you are able to make a model, okay, somewhat, all right. But still, this process is ongoing. Our understanding is not satisfactory from this program, and still the people are keep on working. Okay, so these are like a large area which you can develop your interest. Okay, so in this aspect, you can search like several research articles. You can study. Okay, so how they develop this modeling program and what kind of analysis they are performing. Okay, so between like each modeling program, what's the difference between like each modeling program? Okay, already somebody is making this modeling program and they are like arguing something, and another fellow is coming here and he is making a new modeling program. So what's the difference? What is the link between them? And can I do something here? So you have to think over like this. All right. So what I need to say is that the primary structure, okay, you cannot underestimate. All right, you cannot don't think like primary structure is useless. All right, we cannot use, we cannot get any information from the primary structure. Don't think like that. All right, so it's very important. All right, so I will tell you like another instance. Uh, hormone, right?
Yes, sir.
So what is hormone?
They are basically proteins.
Basically,
Proteins.
proteins. Why you are saying protein? It is not protein. If you say protein, then you cannot say it as a hormone, right?
Okay, sir.
Yes or no?
Yes, sir.
So hormone is made up of what?
Steroids.
Steroids. Hormone is
Sir, lipids.
huh?
Lipids.
Come on, yeah. Hormone is made up of amino acids. Okay. Hormone is made up of by amino acid only. Okay. But the number of amino acid present in the hormone will be very less. Maybe 10 amino acid, maybe 11 amino acid, or 15 amino acid. That's all. It's just a polypeptide. But okay, you are calling it as hormone. Okay. So you know antibody. So antibody is also made up of amino acid. But you are calling the polypeptide as antibody. Why you are calling the polypeptide as antibody? One, yeah.
So they act against antigens.
They are acting against antigen. But it is, you can call it like a protein, no? Yes or
Maybe
no?
so maybe depending on the number of amino acids. Yes, yes, yes.
What's the number of amino acids? Tell me.
I don't know, sir.
Uh, Not like that. Okay? And again, you are calling some polypeptide as enzymes. Yes or no?
Yes, sir.
Right? So we have like a different naming system. Hormone, antibody, enzymes, protein, depending upon the function of the polypeptide. Right? So we are categorizing based on the function of each polypeptide. So some peptides we are calling like hormone or antibody, whatever. Alright? So these functional difference basically arises from the primary structure, from the sequence. Okay, so the sequence dictates the function. Okay, so in human genome, how many proteins are there? Can anybody say? Come on, yeah. In human body, all right, how many proteins are there? No one? Madhavi?
So the exact number I'm not sure. Like
Roughly tell me. I'm not going to blame you. Roughly tell me.
roughly some thousands.
Not thousand, not thousand. It's around like 35,000. Okay. So, so we have a huge number of proteins in human body, right? And these 35,000, okay, approximately, each protein is not of same length. Do you agree or not?
Yes, sir.
They are not of same length. They do not belong same function. So they do not belong same structure. Okay? So what I mean to say is here the diverse structure or the diverse function is basically inherited in the amino acid sequence. So the amino acid sequence dictates the function as well as the structure. So it is not just for the human being, it is for each and every living thing. So every genome is having like thousands of proteins. So each these protein, right? So these protein do not have like a same structure, okay, same number of amino acid, do not have like the same function, okay. So you have like a diversity. So this diversity is basically is caused by the primary sequence. Okay. And then the primary sequence, it is also giving like several information. Okay, so the protein is like polypeptide. It is synthesized in the in the cell, the ribosome, right? So it is keep on adding like one by one, right? And then you have you are getting like a polypeptide. Okay, so the sequence, right? It is giving some signaling peptide, right? So this signaling peptide, what it will do? It will take the protein, the synthesized protein, to the particular destiny. Yes or no? So once it is reached that destination, that this signaling peptide will be cleaved from the original polypeptide. And then this polypeptide will fold into the structure and it will go. Yes or no? So the amino acid dictates, okay, the location of the protein, right? And after reaching the place, the protein may undergo some chemical modification, phosphorylation, glycosylation, right? Some chemical modification is also there for some proteins. Okay, again, this chemical modification, it is informed by the amino acid sequence only. Okay, so the signaling peptide, it may lead the it may lead the protein to the cytoplasm, or it may take to the nucleus, or it may take to the cell surface, right? And like in some polypeptide, it will tell all the polypeptide, okay, does not bind with metal atom, does not bind with prosthetic group, that is heme group, right? Only some polypeptide binding with heme group, binding with metal atom, binding with some lipids, binding with some sugar, or binding with some nucleic acid, right? So how come these polypeptide know that I have to accommodate a sugar molecule? How come this polypeptide knows that I have to interact with some nucleic acid? How is this possible? Only the sequence. Okay, so only the sequence gives the information to the polypeptide. Alright, so the primary structure is like very important and it is giving us like a very useful information. Okay, so how we derive all this information? Because we know like the sequence of like human genome. So we are comparing the each sequence one to one. So we know the function of each protein. We know which sequences that are binding with the sugar, which sequences undergoing chemical modification, which sequence undergoing like a nucleic acid interaction. So all these things that we know, right? So we have like a sequence comparison, we have this chemical information, the biological information. You combine these things, right? So we can make some prediction. Yes or no? So there will be some signature region will be there. Yes or no? Come on, yeah. Do you know what is mean by signature region?
Sir, that is conserved region.
No, your voice is not clear.
Sir, that is conserved.
Conserved region, very good. So conserved region, right? So all this information, based on this information, we can make a prediction. So the sequence itself giving us a lot of information. All right. Okay. So why now we are like somewhat like clear about the primary structure. Okay, Amal Raj, Amarraj, you speak, yeah, don't give the motor. Okay, drr, drr, drr. Only I'm hearing the sound. Drr, drr, drr. Are you in biking?
No, sir, I am here.
Why you are making drr, drr? Speak something.
Sir, my mic, I think there is a problem with the mic, I think.
Now, not with mic, yeah, I know. All right. Tell me, so far I was talking about primary structure or secondary structure?
Sir, primary structure.
Are you sure?
Yes, sir.
What is secondary structure?
Secondary structure, there are two types, beta sheet and alpha helix structure. Based on from the primary structure, the secondary structure is formed basically.
Tell me then, from the primary structure, how you are going to secondary structure?
So when the secondary structure is formed, there are mainly disulfide bonds are formed between the cysteine residues between polypeptides.
There is formed disulfide is coming up. What are you saying, man?
So different arrangements are formed. That is beta sheet and alpha helix.
Hmm.
So from the hydrogen bonding is there in the secondary structure conformation basically.
So what is the hydrogen bond?
Between the oxygen of carboxyl group and hydrogen of amino group.
That is the hydrogen bond.
Yes, sir.
Okay. So here, so can anybody describe some more in detail how you are coming from primary to secondary structure?
Sir, the interactions will happen between the backbone of one amino acid with the backbone of the other amino acid. Sir. Like hydrogen bond will be formed in the main chain itself. So that will give the secondary structure.
Very good.
And I think tertiary structure is like interaction between the side chains of the different amino acids.
Okay, so that we will come later on. Okay, so we are going to see this secondary structure only. Okay. Okay, so Madangi told one useful information is making Madangi and Amalraj also told like useful information. Okay. So there will be hydrogen bonding formation in the backbone of same polypeptide. Okay, so that results a secondary structure. So we know the secondary structure like alpha helix and beta strand. Okay. So before going into this secondary structure, so I want you to know about the hydrophobicity, right? Hydrophobic core of the protein. Okay. So if you take the globular protein or any protein, okay, there will be definitely hydrophobic center, right? The core of the protein will be hydrophobic place, right? So where you will have no water molecule. Yes or no?
Yes, sir.
So you will be having some hydrophobic core, right? Okay. So do we have like any amino acids which is having like hydrophobic side chain?
Yes, sir.
Yes, sir.
Yes. Glycine and iron.
Glycine is having hydrophobic side chain. Glycine, I think there is no side chain at all.
Sir, alanine.
Alanine, valine, and leucine and isoleucine.
Very good. So not glycine. Okay, alanine, valine, leucine, isoleucine. You have the hydrophobic side chains. Okay. Apart from that, is there any other amino acid which is having like a hydrophobic side chain? Methionine. So, do you think methionine is having hydrophobic side chain or chandini? How? In methionine you have sulfur, you know that? So sulfur is hydrophobic or what? Then you are saying methionine is having hydrophobic side chain. Or chandini? Okay, somewhat. Okay, but it is not a hydrophobic amino acid. Come on, any other amino acid?
Sir, aromatic amino acids like phenylalanine and tryptophan.
Very good. So phenylalanine and tryptophan do have hydrophobic side chain. Apart from this, any other amino acid?
Sir, proline.
Proline, you can say like the side chains are like covalently linked. You can say proline, right? Yeah, any other? One, yeah. Any other amino acid? Glutamine. Come on, yeah. Glutamine. Why? Polar amino acid. So what atom you have? Yeah. Okay, so you have some nitrogen. Okay, so glutamine is not hydrophobic amino acid. Okay, you have the nitrogen. Okay, so glutamine is having nitrogen, but glutamate does not have the nitrogen, right? So we can call glutamate as a hydrophobic amino acid, right? What, Chandini? So glutamate is hydrophobic amino acid.
No, sir. It is hydrophilic.
Why?
Sir, it has that COO minus group in it.
Right. So you have the oxygen, right? Okay. So you have some idea about like basic things about that amino acids. Okay, that's good. Okay. But remember, you should know. Okay, you should know. You should be able to draw the chemical diagram of like each amino acid. That's why I keep on testing you. Okay. There is no other way, right? Even now, I'm not sure whether you are really telling or you are just looking the book and telling me that I do not know. But my duty is somehow make you to learn. That's all. I can induce you. That's all I can do, right? And learning is coming from your own interest only. Okay. So learning and getting expertise, it is coming from your own interest only. We are just a catalyst. The teacher is just a catalyst. We will ignite the flame. That's all. Okay. So you will burn everything only by the zeal that you have, only by the interest that you are developing. All right. Okay. So what I wanted to tell you is we have some amino acid which is having the hydrophobic side chain, right? So these hydrophobic side chain definitely will not like to float into the surface of the protein because the surface of the protein is exposed highly to the solvent molecules, right? Highly to the solvent molecules. So the hydrophobic side chains will be facing towards the core of the protein, towards the center of the protein, or it will be trying to keep themselves away from the water molecule. So it will make a hydrophobic core. Okay, and similarly, you have like a glutamic acid, right? And you have like aspartic acid, you have like asparagine, you have arginine, all these charged molecules, charged amino acid, right? Whether they have like a nitrogen atom or like oxygen atom or like sulfur atom, which is carrying like positive or negative charges, they will not like to present in the hydrophobic core, right? So they will be trying to be exposed in the solvent molecules, right? They most of the time they will be on the like the surface of the protein. So in the protein you have like a different layers. So inside you have hydrophobic core. So outside you will have hydrophilic surface, right? So from the center to the outermost area. So imagine as a circle, a globular protein or whatever, like different shape of the protein. Remember as a circle. So from the center of the gravity, the hydrophobicity will be keep on decreasing when you go outside. Okay, or from the center of gravity, when you go outside, you will keep on increasing the hydrophilicity. Okay, so this is the like overall image of like almost for all the proteins. Okay, so the hydrophilic amino acid, the side chains, okay, it will be interacting with some solvent molecules, some metal atom, or whatever. And hydrophobic side chain, they will interact among themselves, but not with the charged atoms. So it is making some hydrophobic core for each and every protein. Again, this information is buried in the primary structure. So the primary structure dictates which are all the amino acids to be present in the hydrophobic core. So which are all the amino acids to be present on the surface of the protein. Again, this information that is coming from the primary structure only. Okay. So I'll stop here. So I have given like a basic idea about the primary structure and secondary structure. Maybe in the next class we will continue this. All right. So if you have like any doubts so far, you can tell me. Otherwise, we will wind up the class.
Sir, I have a confusion, sir.
Tell me, Sadia.
Sir, as far as I know, hemoglobin structure has two alpha chains and two beta chains. So how can it be a homo polypeptide?
Yeah, it's a heteropolypeptide. So that is like when I was telling you, it is I told you it's a homopolypeptide.
Yes.
Yes, so there is a slight change in the sequence between alpha and beta chain. You are correct.
Okay, sir. That is what I wanted to ask.
Yeah, any other question? Okay, so there is no more questions, so we'll stop here. But I have a question for you guys. So if anybody tested positive, Chandini, Amarraj also.
Luckily, no.
Amarraj, you are driving scooter again. Speak something, yeah.
No, sir. I'm not tested positive actually.
Okay, what about other students?
No, sir.
No, sir.
Okay, Chandini, where are you? Where from you are? Chennai, okay. So you are taking care of you. So daily you are taking steam. Okay, so take care of you. So if you have like fever, you have to continuously take the like Dolo 650 or whatever. Okay. Uh, so I am telling for general for all the students. Okay. So it's very like difficult times for us. So you know what happens in north. It is slowly migrating. That is like not slowly. It's very fast. Okay, very fast. People are expecting to the third wave also. So the third wave is going to be like serious, and even it can affect the children as well, kids also. So it's like very tough period. So I'm giving you like information unnecessarily. Stop roaming around your place. Okay, stay home, stay safe. All right. Take care of you, Chandini. Okay, so we stop the class right now and maybe we'll see next week. All right. So tomorrow I will not take the class. So tomorrow you can call any other professor to utilize my that class timing.